All right. So the first thing I want to do is I want to go over one problem on test 2 that everybody everybody got wrong. Well, not everybody, like 90% of people got wrong. So I just want to take a look at that. All right. And so it was this problem where we're looking at alpha helices on a hydrophobicity plot. And like, I want to say 90% of you said the right plot. And the question is, which plot is likely to contain alpha helices that cross the membrane? All right, so for a hydrophobicity plot, positive numbers are hydrophobic. Negative numbers are hydrophilic. Now the membrane itself is hydrophobic on the inside. So if I'm an alpha helix that goes through the membrane, that alpha helix must also be hydrophobic, right? So what you're looking for is you're looking for large areas of hydrophobicity, and that they will repeat, because what happens for a protein is that it'll go through the membrane, come out the other side, and then go through again, come out the other side, go through again. And so that's why it's the left plot where you would have 1, 2, 3, 4, 5, 6, 7 different helices. So each one of these is an alpha helix, right? And we can tell it's going through the membrane because it's very hydrophobic. So that's just the problem I wanted to cover because, yeah, just about everybody got that wrong. Other than that, there was no problem that like the majority of people got wrong. And so what I've done is that I've already dropped your lowest scoring test, and that will update as you do test 3 and test 4. So when we do test 3, I will look at all your tests again, and then in Blackboard, the lowest one will drop. So the grade you see on Blackboard is currently what your grade is in the class if we drop the lowest score on the test. Since it wraps around, I was thinking since it wraps around and kind of goes outside the membrane for a bit, that would go from hydrophobic membrane to hydrophilic. Yeah, that's what it's doing here, right? Hydrophobic, then it drops back down because it's hydrophilic. Hydrophobic drops back down. Hydrophobic drops back down. Remember, this is a running average, which means that like it's not like simply, oh, this amino is hydrophobic, so it gets a score. These peaks are, we're just looking at windows, right? We're looking at like what's around it, and we're moving our windows as we score. So that's why, I mean, it doesn't drop all the way down. How is it not the one on the right? Because the one on the right doesn't have like hydrophobic peaks over and over and over again. No, but yeah, below 0 is hydrophilic. So I want hydrophobic, right? It's only outside the membrane for a couple residues, not a lot of residues. So if I'm doing an average, it's not going to like drop to like minus 30 because it's still averaging in the hydrophobic residues around it. So that's why you see it drop and like maybe touch 0, but that's all it does. No, in the video I explained it, mine should look like the one on the left. The one on the right is a regular folded protein. That question was extremely confusing. I thought it was pretty clear. Which plot has alpha helices that cross the membrane, where it's, if you're crossing the membrane, you're hydrophobic. What does the hydrophobicity index show you? It shows you which residues are hydrophobic, right? Yeah, I did actually go over this exact question in lecture, like 100%. I don't think it was the exact example, but it was basically the same question I asked. How do we find part B? You just count the peaks. There's 7. Yeah, I think I use a different plot on the test, but I might have asked the same question where I hand drew the plot. My plot would have looked like this. Whoa, 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 right? So that's what my plot looked like. So here, we don't go quite as negative, but you see these giant peaks. What this is saying is like, it's just not hydrophobic or hydrophilic. They just keep switching. Why am I yelling? Okay. If you want to show me the plot and it looks more like the right, I would highly doubt that's what I drew. If you want to take a screenshot and it doesn't look like the plot on the left, I will apologize. But I'm pretty sure I just drew what I drew again, right? I'm pretty sure my plot looked like this, which if I'm trying to squint and I'm saying, which one does the one I drew look like? Does it look like the one on the left or the one that looks like on the right? You're going to be hard pressed to say that it looks more like the right than the left. It looks more like the right. You might need to get your glasses checked, right? Because it goes peak, 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 peak. On the right, we don't have that. On the right, you have maybe this, 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 but the rest is just, and here we have nice up, 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 up. So that is what's going on. You found the slide. Looks to the right. All right, I'm going to pull open my slides right now. Or I'm going to open up the answer key. How about that? Because I have the answer key on this website as well. And you can say it looks like the right, but I do not think so. What date was that? What date did we go over that? Like what problem set? Hydrophobicity. Hydrophobicity. Where is my plot? Okay, I found it. Share screen. So in my plot, I do go further in negative. I will give you that. But you can see it's constant. It's a constant wave, right? Peak, down, peak, down, peak, down, peak, down, peak, down, peak, down. So it goes up, down, up, down, up, down, up, down, up, down, up, down, up, down. Let's go back to our test now and see which one goes up, down, up, down, up, down, up, down, up, down, up, down, up, down. Right? So here we are back at the test. Well, if you're looking for the switch past zero, well, let me get the test back up, right? If you're going to make that argument that you're looking for the switch past zero as much as I did it on that answer key, I'm going to argue the right only does it like maybe twice, right? Maybe here, let me get my pen out, all right? Maybe here and maybe there. Here, up, down, 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 right? You're alternating because you're going through the membrane, outside the membrane, through the membrane, outside the membrane. Here it's zero. The average of this is zero, right? What was the average for the class of 71? All right, so I'm going to stop arguing this because one of the graph I drew, you can make the argument that, well, I was looking for the thing in the negative. I'm going to say on the right, there was no giant positive peaks as well that would go through the membrane, right? What this is is that it's a folded protein where you have, a mix of stuff. Here, you have giant hydrophobic. I thought it's above 0 and below 0, though. I mean, what's more important is that you have, okay. So this, right, corresponds to this. Okay, this corresponds to this. If you're getting hung up on, I'm looking for above 0 and below 0 and equal, that tells me that you're more memorizing what my chart was and not so much learning what is this graph telling me when I look at this. Because if you learn that, what you're saying is if I'm above 0 a lot, I have a larger hydrophobic residues. If I'm below 0 a lot, I have a larger hydrophilic residue. Right? So only one of those plots is above 0 a lot, right? That's how I'm going to say that. Okay, that's 15 minutes over one question. That's hardly worth anything on the test anyways. So we're going to move on to our next part. If you have any other more complaints, please direct those to my email and we can take them up there. I will send a strongly worded email. If you're going to send me a strongly worded email about question 14, there's probably not an argument that you're going to make to convince me that I'm wrong and you're right. You can try. You are more than welcome to try, though. Remember, this is a dictatorship at the end of the day. If I think I'm right and you don't have a logical argument to say I'm wrong, then I'm going to go over my logic versus your logic. I say that the drawing has big peaks. Again, I showed you a different picture. I showed you the picture that would correspond to. If you completely understand what that graph shows you, right, then you know how to read that graph. You know membranes, yet you pick the chart that's always at 0, which means that you're alternating hydrophilic, hydrophobic, hydrophilic, hydrophobic, hydrophilic, hydrophobic. B says like every other residue is either hydrophilic or hydrophobic. They switch. That's impossible in the membrane, right? You cannot have a hydrophobic residue in the membrane unless it's interacting with another hydrophobic or hydrophilic residue. Yes, let's just drop it. It is alternating. Yeah, that makes no sense in the membrane. Why would you have a hydrophilic residue in the membrane? That makes 0 logical sense. Yes, I have spent way too much time arguing about that. That's why I ask, if you want to argue, please email me so we don't waste everybody else's time, right? All right, moving on. Duplicate. So we were talking about the immune system. And we're talking about antibodies and all that good stuff, right? So we talked about IgGs on Wednesday, and we talked about how we have a site that's variable and a site that's non-variable, right? So we have this purple part, which is variable, and this yellow part, which is constant. This purple part can be like anything, can be like any amino acids, and this is what's binding to our antigens. And this is why our antibodies can bind to like almost anything that's foreign, is because they constantly are changing. Okay, enough chatter about that. I'm being serious now. If chatter continues on that problem, I will start muting people. Again, done, over. That's a one-on-one conversation if you want to have an email about it. I've just given the warning for muting, right? Warning first, and then we'll move on to actually muting people if need to. So case closed, no more public discussion about that. All right. So we talked about light chains and heavy chains, right? How, we did this problem on Wednesday where the antibodies are all connected through disulfide bonds, right? And they have a certain weight. Light chains are 23 kilodaltons. Heavy chains are 65 kilodaltons. And to get these separate chains, light chains and heavy chains, we have to break disulfide bonds. Disulfide bonds are all connected via cysteines. And so if we did various experiments, what would they come out like? So we already did that one. We talked about, this was the very last slide actually, the IgG fold, or IG fold rather, the immunoglobulin fold. These antibodies, they form beta sandwiches, which are just these anti-parallel beta sheets. They're connected by disulfide bonds. And when we bind our antigen for IgGs, they're called divalent, where we bind two things at once, right? Other antibodies can bind multiple, more than two. So we have antibodies that can bind 5 things at once because they have 5 variable regions. But just know that we can bind multiple things with our antibodies. And when we do that, we get this like giant conglomeration or lattice rather, cross-linked lattice of our antibodies and our antigens. And that's how the immune system generally works, is that we clean it up. IgM is the pentamer. Yeah, I believe so. That binds more than 2. And all the different antibodies, they usually react to different things, and we have different levels of them in our system. Like, like we have ones against fungus and all that good stuff. All right. So I'm probably going to answer some of these questions just to move us along here. If I say something that is confusing, please do stop me and feel free to ask for clarification here. All right, so we have variation in the variable loops, VL and VH. That's like where all the variation occurs. But why don't we see variation in the constant region? And here I'm really getting to the idea of like, how does an antibody actually work, right? So the reason we have the variable regions is so we can bind to any antigen. All right, so we need that to be variable. But we also have to recognize our own antibodies, right? And so when our antibodies are stuck onto something, we have to recognize, hey, it's stuck onto something. And so the way we recognize our own antibodies is through a constant region. So we want our own bodies, our own cells, to recognize these constant regions. So wouldn't it be super convenient if the part of the antibody we want to recognize doesn't change? That means we only need one binding site to recognize our own antibodies. So let me go back just a couple slides. Right, so we're talking about this whole how this works. And so you can see we have our B cell and our B cell receptor, and we have antibodies that can glom onto our beta cells or are attached to our beta cells. So where they attach, if you look, they're attached right here on the Y. That's our constant region. That's where we're interacting with our beta cells. So that needs to be constant. Otherwise, we would need different binding sites for every single antibody we had, and that would be a giant pain. It wouldn't be very efficient. So to get around that, that's why the constant site stays constant. Five subunits, 10 binding sites in total. Yeah, so for our other classes of antibodies. But yeah, we're just going to focus. That's all. Yeah, we did cover that in the videos, but right now I'm just going to talk about IgG. All right, so that's why there is a constant and variable region. Questions about that idea? Why that site remains constant? That's basically what the question is. Why does that site remain constant? All right, so if there's no questions about that, why do antibodies raised against native proteins fail to bind corresponding to nature proteins? All right, so the idea behind this question is just to get you to further understand how protein-protein interactions work, right? Where it's all based on charge and geometry. So binding based on charge and geometry. So if I have an antibody that binds to a folded protein, the folded part, so this is folded, has a specific geometry that I'm looking for, and around that has specific amino acids. Well, if I become unfolded, I become like a spaghetti string again, right? That's what an IDP basically is. And if I become unfolded, I lose my specific geometry. Let's say, that's the point I'm binding to, the little circle I drew. Maybe it corresponds to right here now. I lost that geometry, and it might not even have the same amino acids around it, because when proteins fold, they bring amino acids from different regions of our primary structure into close contact. So maybe in my folded protein, let me get a different color here. Maybe in my folded protein, this part of the protein is also near here, and that's what my antibody is recognizing. But in an unfolded protein, my antibody cannot recognize that. So antibodies are extremely specific, and if antibodies will bind to a protein, they will usually only bind to one form of that protein. That is, do they bind to the folded protein? And that's what it usually is. Because if a protein's unnatured, this changes so fast, the geometry is constantly changing, that it's very hard to recognize that for an antibody, because it really needs some kind of like solid structure. All right. So questions about that idea of protein-protein binding, what an antibody is looking for, anything like that? So it binds to two things. It binds to the local structure and whatever amino acids are by that structure, right? So in a folded protein, when you fold a protein, you can bring different parts of the primary structure together that aren't close on the peptide chain, but when you fold, right? So like in my example, I'm bringing this part of the protein and this part of the protein together. So maybe the red part's like right here. And so my antibodies recognizing that. So antibodies are recognizing a specific geometry in either charge or polar, nonpolar. So I guess that's the short answer. They are looking for specific geometry in specific either polarity or charges. When a protein unfolds, it loses the geometry and loses that local charge structure. Right. So let's say like if I bound to an alpha helix, if I'm unfolded, I'm no longer an alpha helix, and it looks completely different. So I can no longer bind to that. That's probably the simpler way to say it. When a protein is folded, it's like the complementary leggings that can snap in. Yeah, denatured, it's a noodle. I don't know what complementary Legos. That makes a lot of sense. I thought you were going to talk about some kind of fashion trend that I was not aware of, which would not be the first time. I'm not a very fashionable person. Yeah, so denatured is wet noodles, wet spaghetti. That's how I think about it. Becomes natured. You fold that wet noodle into actually origami is a great example of this. When you make a piece of paper, it's just like a piece of paper, and then you fold it into an origami like swan. That's your folded protein. Kind of. Any metaphor works. All right, and so question 4, again, was just more review on what we said earlier. And since it's been almost a week, we can briefly go over this, right? So remember how I said at the very beginning here again, when we have our antibodies binding, they're forming this giant meshwork of antibody and antigen, this complex network. So let's say that I have an antibody and I raise it against an antigen. And usually when I expose my antibody to this antigen, we have a precipitate. However, there are certain scenarios when no precipitate forms. So scenario A, we don't have a Fab, so we only mix Fab fragments with the antigen, all right? So let's take a look at what the Fab fragment is, or FAB really. So the FAB are this part, and the FAC, the crystallization, let me use brown. I don't know why I used brown and dark red. That was a poor choice. Anyways, the FC is the stalk. So if I had, right, just the FAB regions with my antigen, there's no way I can form this meshwork because I don't have any stalks or FC regions to, let me switch my colors again. I don't have any FC regions to hold my two binding sites together. So it's like I'm releasing just one crab claw to go and attack my antigens. You need both crab claws or both FC regions or FAB regions. Talking too fast here. You need both FAB regions attached to your FC region to have binding at two different antigens and have it linked together. What question or part A is saying is that imagine I just take away that blue stalk, right? And I just have, all right, I remove all the stalks. I can't form this network because I'm no longer holding two things at once. Only one thing at once binds. Does that make sense? Questions on that explanation? All right, for B, let's say I have a small antigen. Why can't I form this precipitate? If the antigen's super small, right? If it's super tiny, it probably won't have multiple sites to bind. So to get a precipitate, you need multiple binding sites on the same binding sites on the same foreign molecule, right? If it's small, it probably won't because there's not enough difference in geometry, right? So if my a good example of this is like a sugar. If I'm looking for a specific sugar to bind to, there's no other way that sugar can be different. And so I probably won't have a precipitate because I need at least two antibodies to bind to the same antigen at once. If it's really small, there's no way I'm going to have two antibodies bind at once. Questions about that idea? All right, and for C, let's say I have a lot of antibody and few antigen. Why won't I form this? The precipitate, yeah, is just a network of antigen antibodies where you get so much of it, it kind of crashes out of solution because it can no longer be dissolved like in the blood. It's just that giant web. Yep. Right, so C, let's say I have my antibody in great excess over the antigen. So if I have like 1 million antibodies, like let's say I have 1 million of these, and I have 10 antigens. Why won't I form this web? Probably because if we look back at our picture, we need 2 antibodies, or we need 1 antibody to be binding 2 things. 1 antibody binding 2 antigens to get this precipitate. If I have so many antibodies, the chances of, if I throw 1 million antibodies at 10 antigens, the chances of 1 antibody binding 2 things is so small that it won't happen. And so you'll have like 1 antigen and it'll just have, 3 antibodies on it. But those antibodies won't be connected to a different antigen because that other antigen will have all its binding sites occupied by a different antibodies. So we're not going to form this nice web up here. And it's the same idea. If I have a ton of antigens, but very few antibodies, again, even though I might bind, each of my antibodies will bind 2 things at once, there's not enough antibodies to form this connected web. So you might have you will have 2 antigens held by an antibody, but you don't have enough antibodies to keep with that web going. So really this question is really getting at how to get that precipitate. All right, questions about either A, B, or C, just understanding how our antibodies are working here in the cell. Can a precipitate form without constant regions? Like the AG is bound to 2 Fab fragments, but no constant part. That's kind of like what A is saying. And it's not possible unless something is holding those 2 Fab fragments together. So it would, how would that work? Yeah, you would have to have this bridge here. As long as you have that bridge that connects them, a precipitate should be able to form. But the constant region is really holding it together. So if I chopped off all of the constant region like that, nah, you're not going to form a precipitate because you're not holding your antigens together. You'll just have your antigens still being free floating with just like little variable regions attached to them. Does that make sense? Okay. The last thing I want to talk about in this slide briefly is the autoimmune diseases. And I'm sure if you've heard about these, the most common one being arthritis, which attacks your connective tissue. And what these are is that your antibodies are supposed to only bind to foreign material. However, in autoimmune diseases, your antibodies have now recognized you as a foreign material by accident. We don't quite understand how this works, why your antibodies just decide, oh, like for arthritis, oh, your connective tissue is now foreign. Let's bind to it and destroy it. Because what usually happens is that while you're developing, right, any B or T cells that recognize your body as foreign, they usually get destroyed naturally before they are matured. And this happens as you constantly grow, right? Because you more or less randomly make antibodies. Your DNA just goes through random mutations and you are constantly spitting out antibodies. And there's a chance, since we have like 10,000 molecules in our body that we need, or more than 10,000 actually, there's a chance your antibody will say, hey, I found something, which is really you. Usually we can eliminate this, but sometimes we don't. Again, not really understood how that works, but it does happen. And we see this happening when there's injury and disease. So if a part of you get injured or it gets infected, right, it might, since you're throwing a lot of antibodies towards that region anyways, maybe an accident happens and whoops, I targeted myself. So that's basically what an autoimmune disease is. That they become overactive. Okay. So a naive beta cell is mature and they become overactive. Okay. Like I said, those of you who have taken immunology classes probably know more about this than I do. I'm more on the antibody binding stuff thing. But, and I'm going to go over this super fast. There is a disease, systematic lupus erythematosus. I do not know if I got that right. I probably did not. SLE, which means you make antibodies that attacks DNA and phospholipids, which is super bad because in question A, why don't you want to do that? Because everybody has DNA and phospholipids. Every one of your cell does, right? If you look at different organisms, we basically all have the same DNA. So you're going to be attacking everything. And what happens, B, why does it happen? Why do you have a lot of this in your tissue? Because your tissues have phospholipids and DNA, right? So those individuals with SLE, that is very detrimental when you are more or less just attacking your own DNA or anytime a phospholipid is presented. But yeah, so the point of those questions is just talk about, again, understand how an antibody works. It's looking for a specific thing. Autoimmune disease is your antibodies start attacking yourself or start recognizing yourself as foreign. So if there's any questions about that, please let me know. Otherwise, we are going to continue to motor on. Try to keep on moving here. And I don't want to do that. One sec. Didn't show up on my screen. Yeah, there we go. Okay. So we are done with proteins for a little bit. Those of you who hate proteins, rejoice. We're done for them for like a chapter. Then we're going to go right back to them. But we are moving on to a much sweeter topic. Ha, 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 ha. I did all the laughing for you. Sugars, aldoses, and ketoses. All right. So unlike the amino acids, I don't have that many sugars for you to memorize. So those sugars that I have in boxes, these are going to be the sugars that are most important for biochemistry. So these are going to be the ones I want you to know. So it is going to be like how I tested you on the amino acids. I will give you a picture and I'll ask you to name it. Or I might give you a name and then multiple images and ask you to tell me which name goes with which picture. But there's only 1, 2, 3, 4, 5, 6, 7, 8 different sugars for us to know. In our sugars, we have two major types that we're going to fall into, aldoses and ketoses. So the difference is for an aldose, the carbonyl carbons at the number 1 carbon, and for a ketose, this is at number 2 carbon. And the way we generally name our sugars is we just say is it an aldose, then we name how many carbons are in it. So this is an aldotriose. This would be a ketotriose. Aldotetroses, tetroses. This is a keto tetrose, so on and so forth. So what I might also ask you is I might give you like a random sugar, and I won't ask you for its name, but I'll ask you for the class. Be like, is this an aldopentose? Is this an aldotetrose? Is this a ketopentose? Something like that. All right. So questions about what I'm asking you to know in terms of like pure memorization for this slide. The difference between the bottom 3 aldoses, the one that I have boxed. Sure, we're going to actually talk about that in the next couple of slides. So let me hold off on like what the difference is. The tetra and the penta and all that correlated to how many carbons are in the spine. Yeah, right. So pentose, 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Tetros, tetra, 1, 2, 3, 4. 1, 2, 3, 4. Structure, class, and name. That's correct. Yeah. So let me talk about the differences in these sugars, right? The only difference, the way we call let me back up there. For these different sugars, all that's different is the position of their OHs, right? So that's a good way to like actually memorize these. So for glucose, right? If you want to memorize that glucose is an aldohexose, you already have part of the structure. If it's an aldose, that means it's C double bond O. Then if it's a hexose, it has 6 carbons. So 1, 2, 3, 4, 5, 6, right? And H2OH. The only difference between all of these sugars are which directions the OHs are pointing, right? Because these OHs are, they have directionality. Where we're showing right and left, you can think about that like above the plane and below the plane almost. So one way to memorize glucose is it's right, left, right, right. Right, left, right, right. OH, 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 OH. While mannose is left, left, right, right. So mannose is LLRR. So 1, 2, 3, 4, 5, 6. Double bond O, left, right. Oh, left, left, right, right. I did glucose there. So that's all that's different between these box structures, are what directions the OH are pointing on the carbon skeleton. We have special names for these, right? So if we look at glucose, glucose only has 4 chiral carbons. Where remember we talked about this at the very beginning, basically a chiral carbon is attached to 4 different chemical units, 4 different chemical or molecules, more or less. So carbon 2 has an H, OH, C double bond H, and the rest of the tail, right? So it's a chiral carbon. So carbon number 1 is not chiral because it's not connected to 4 things. Carbon number 6 is not chiral because it's not connected to 4 unique things. It's connected to 2 hydrogens, right? So if we look at all of our aldohexoses, we have 16 different possibilities. That is, a chiral center can be the OH facing the left or the right. So we have two possibilities, 4 positions. We have 16 different possibilities, and we call these stereoisomers. A stereoisomer is a sugar with the same number of carbons, and let's say they're all aldoses, right? It's just how many different possibilities you are. So if we go back to the previous slide, glucose and mannose are stereoisomers of each other. Same number of carbons, both aldoses. The only difference is which directions our OHs are changing. So everything on the bottom left there, they're all stereoisomers of each other. All right. And when we are looking at sugars here on Earth, the majority of them are D sugars. What a D sugar is it has the OH furthest away from the carbonyl carbon. So the OH that is furthest away, that's still chiral, that's going to be on the right. So again, I'll repeat that. The chiral OH furthest away from the carbonyl carbon, if it's pointed to the right, that's a D sugar. Almost every sugar we're going to look at is D, but there are L sugars. An L sugar would be, well, OH is on the left. So if it's on the left, it's an L. If it's on the right, it's a D. D for dextrose, not dextrose, but it's Latin for right, basically, what that D stands for. To further complicate things, sugars can also have what are called epimers. An epimer is when only one chiral carbon is different. Our example here is glucose and mannose, right? So if we look at glucose and mannose, the only difference is C2. C3, 4, and 5 are the same. So if you only have one difference, you're an epimer. But they're also stereoisomers. So it's one of those word puzzles. Every epimer is a stereoisomer. Not every stereoisomer is an epimer. We have OH on the left side of carbon 3 of glucose. Does that mean anything? Nope. It only means, we only give it a special name. Well, it means something in that it's a different sugar, right? So based on the OHs that are on the right and left, every combination we have like a different name for. But we only give it the designation D or L if we look at the last carbon away from the carbonyl carbon. That's the only time it gets like a special letter designation. All right, any other questions about epimers or stereoisomers? All right, I did want to jump into one question. I know I'm over time, but one question just to check your understanding while this is still fresh, right? So I have a ketopentase, ketopentose, ketohexose, and ketoheptose. Based on that, and based on the little math I just did, how many stereoisomers are possible for a ketopentose, a ketohexose, or a ketoheptose? We didn't talk about heptose, but heptose is 7. So 7. Hexose means 6 carbons, and pentose means 5 carbons. So see if you can calculate that really quick, and then we'll call it here. $25, $36, $49. too high. Too high. Let me show you the ketopentose, right? So pentose means we have 5 carbons. Keto means the second carbon is our carbonyl. So this is CH2OH. This is CH2OH. OH, not OS. So we have 2 chiral carbons. Chiral carbon right here, chiral carbon right here. They have 2 possibilities, left or right. So you have 2 possibilities raised to the power of how many chiral centers you have. So for a ketopentose, that's 2 raised to the power 2. That's 4. For a hexose, 1, 2, 3, 4, 5, 6. Not OH, sorry. H2OH, H2OH. So I have 2 possibilities again, because it's either to the right, the OH is either to the right or to the left. I have 1, 2, 3 different chiral centers now. So that's 2 raised to the power of 3, better known as 8. And a heptose, 1, 2, 3, 4, 5, 6, 7. Keto, so it's right there. H2OH, H2OH. 1, 2, 3, 4. 2 raised to the power 4, which is better known as 16. So it's 4, 8, 16. And you can follow, and so if we added one more, keto octose would be 32. Nonose, which don't exist, but that's 64, so on and so forth. All right. Any questions about that? If not, that's what we'll end it. Looks like we do. Will they all raise to a multiple of 4? It's more a multiple of 2, to be honest, because it's base 2, right? So it would be 16, 32, 64, 128, which is a multiple of 4, but it's a multiple of 2. 128, 356. Then I can't do that in my head after that. But yeah, it's just 2, 2, 2, 2. Same thing for aldoses. Exactly. The only difference with an aldose is where we put the carbonyl carbon, right? So for a aldotetrose, we have 2 carbon centers, so it's 2 raised to the power of 2, which is 4. For an aldopentose, here 1, 2, 3, 4, 5, we have 3 chiral centers, so 2 raised to the power of 3 is 8. Same exact. Yeah, for sugars, it's always 2 raised to the power of how many you have, right? So, I mean, and that equation is general for anything that has 2 possibilities. So like flipping a coin is either heads or tails. So if I flip a coin 2 and I flip it 5 times, I have 32 different possibilities. It can be all heads, 31 heads, 1 tail, so on and so forth. Yeah, that's just a common probability equation there. Yeah, so for sugars, 2 raised to the power of how many chiral carbons there are. Yep, that's how many different sugars you can be, or how many different stereoisomers. All right, so that is all the time I have for today. I'll put this video up. I will remember to put homework up this time. I'll make sure to do it this time. So you got 1 freebie there because I had 2 tests that week and forgot, but never again. All right. As always, if you have questions, let me know. And I will see everybody on Wednesday, hopefully, or Tuesday. Take care, everybody.